Today we will be discussing their lab protocol article in PLOS ONE and associated protocol in protocols.io with doctors Sebastien Rodrigue and Antoine Champie. Sebastien is a professor in the Department of Biology at the Université de Sherbrooke. Sebastien and his team devised innovative strategies to engineer bacterial genomes and tackle various diseases. He is particularly interested in minimal organisms, synthetic genomes, and live biotherapeutics. Sebastien is also a co-founder of Tatum Bioscience, a biotechnology startup developing new therapeutics using synthetic biology. Dr. Antoine Champie is a postdoctoral fellow at the Rodrigue Lab, where he does synthetic biology, including genome edit design and engineering. He has started his academic career in France as an undergraduate student in microbiology. During a year-long student exchange experience in Sherbrooke, he met Professor Rodrigue. Under his mentorship, Antoine pursued first a master's, then a PhD, both in synthetic biology. Antoine's research focuses on DNA manipulation, genome and plasmid design and assembly. He also performs protocol design and optimization, extending into bioinformatic analysis, including genome sequencing and transcriptomics. In addition to wet lab skills, Antoine has expertise in electronics, programming, 3D modeling, and 3D printing. The article we will be discussing today is entitled Enabling Low-Cost and Robust Essentiality Studies with High-Throughput Transposon Mutagenesis and the Associated Protocol in protocols.io. Thank you, Sebastien and Antoine, for being here with us.
Thank you for the invitation. Thank you.
So to kick off, can you please describe briefly what the protocol is about, how to use it, and why it is relevant?
Sure. The protocol is about transposon mutagenesis. And the idea is to bombard cells with transposons, which are little pieces of DNA that can be inserted at random in a genome. And when they're inserted, they disrupt the gene, and the gene's activity will no longer be performed. And this enables studies to know which genes are essential or important under different conditions.
And the way we use this protocol is first you have to have a target cell, for example, a bacteria like Escherichia coli, and you mix a specially designed donor strain with this recipient cell. You let them conjugate on something we developed called agar plugs, which is what enabled us to have a really high throughput, higher than what existed previously. And once you've let the donor strain conjugate the transposon toward the recipient cell, you've got a mutated population that you then select using selective passages in selective medium. And you get a mutated population, which you can then harvest the DNA and sequence using Illumina sequence.
So to understand the significance of the protocol and its optimization, can you help us understand what was the state of the art at the moment when you decided to work, and how your optimization led to this high throughput mutagenesis?
Sure. Transposon mutagenesis is not something new. It's been performed for decades. So people have been working on this for a while, but usually with a relatively low number of samples. So usually when people compare a few samples or maybe a dozen of samples together, it's already considered a relatively large or complete studies. But in our case, we had two motivations at the beginning. we wanted to make sure that these results were reproducible. So we wanted to increase the number of replicates so that instead of comparing 2 or a few samples, we could have a lot of data supporting very strong and robust statistics. And the other reason for doing the protocol was just to increase the throughput so that larger projects, very ambitious projects with lots of different strains could be undertaken and perform in a relatively short amount of time and with reasonable resources.
Yeah, the project came before the protocol. We wanted to do transposon mutagenesis on thousands of samples, and then we started to work on how could we realistically perform this transposon mutagenesis, and that's how the protocol came to be. We had the project, and then the protocol came to enable it.
So you had the project, you needed to improve the protocol to be able to execute and undertake that project. What was then the motivation to, since you invested this time to post it in the platform in the first place, and second, to have it peer-reviewed as a standalone publication?
Yeah, I think we have all read papers or articles where we see a nice method, and then we try to implement it in our labs. And we realize that there are so many small details that are not mentioned because of the lack of space, for example, or because it's maybe not the best medium to give all these details. So we wanted to share the protocol with all these information so that people could easily use the same method that we developed without having any headaches and trying to figure out small details and optimize everything. The second part of your question was why have it peer reviewed? And I think there were 2 main reasons for this. The first one was just to make it public and have peer validation that our method was sound, so that it would be accessible and would be accepted in the community. And the second one was that because we're interested in larger and very ambitious projects, we wanted to publish the method first so that the next projects would focus on the biology and the conclusion instead of validating the method for half of the paper. So for us, it was really a good way to provide information, but also publish it and put it a little bit behind us so that we can focus on other things on the upcoming publications.
Yes, and we also think that on its own, the protocol can be really interesting for people who will not necessarily care about the big project we intend to do with it. So it was a nice way to put it available for many people, not just people who would be interested in what we will do later with it and then stumble upon the protocol. It was a nice way to have it like more accessible and not as linked to our other research as it would have been if it was published alongside the main result. So accessibility.
Yes, and having this, modular concept of communicating your science. I mean, not only the biological question, but the method development should make the method itself more discoverable and more reusable as an independent module, right, of your research.
Exactly.
Yeah. Could you elaborate then in how the feedback that you received from the peer reviewers helped shaping the final version of the protocol?
I think overall the protocol didn't change that much after peer review. There were some points that needed clarification, so we made sure that every little detail was really easy to understand and follow. Sometimes when you write something, you're so much into it that you know what it means, but other people reading it will not necessarily get the same idea. So I think that was really for clarity. People, the comments that we got, I think, are improving the paper so that I think anyone can read it and understand some of the most important concepts.
And what was the motivation to get the peer review history published together with the paper?
I think it's more like as a principle, we want our research to be as open as possible. So we've got nothing to hide, so we won't hide anything. Just why not? I think the question is, why would someone want to hide the peer review process?
Okay, that's a good and fair point. And thank you for being so open, regarding your science. But I would like us now to dig a little bit into the protocol itself. For what you have described, it seems that this protocol will be or is already the foundation of many projects to come. And I would like to know if while optimizing the protocol, was there anything that you tried that didn't work and didn't make it to the final version of the protocol?
Of course, a large number of things, unfortunately, but that's science. The tools that come most easily to mind are first, we want the current protocol uses what's called solid conjugation. You mix your donor and your recipient strain. You let them dry onto a solid medium. In our case, LB agar.
Yeah, and just to make sure that everyone can follow. So at the beginning, I mentioned that we bombard cells with transposons, but that's more like an expression. So what we actually do is mix two populations of bacteria. One is the target, so the strain that we want to mutagenize, and we have another strain which contains the transposon DNA on the plasmid, and there will be a transfer from the donor cell to the target cell. So the transposon plasmid moves from the donor to the target, then integrates into the chromosome, and we get mutants. So that was just the...
Thanks for the elaboration.
The smaller details for people to follow.
And the process of conjugation, which brings the transposon to the recipient cell, can happen in solid conjugation, which is what I described. You put the donor and the recipient cell on a solid medium and you let them conjugate. But it can also be done in liquid condition, when you just have to mix the two populations in liquid condition, in liquid LB. And it's a much faster way to perform conjugation at the cost of efficiency. And the original design of the protocol was to use this liquid conjugation to bring the transposon to the recipient cell. But despite the liquid conjugation functioning, it never could work properly with the transposon. And it was the first big failure of the protocol, but we pushed through and managed to use solid conjugation instead, which is like the main, one of the first things that didn't work. And the other hurdle, which is still a bit of a mystery, where science and magic and magic mix a bit, because at some time the optimization was performed during the COVID pandemics, and we had a difficulty to acquire some specific plastic deep well, which we use for conjugation, and with no explanation, with one brand of deep well plate we use for conjugation, everything worked flawlessly. And suddenly, when we had to change supplier, everything stopped working. And I think we never figured out what was different between the two plastics, but it's a few months lost trying to figure out why it suddenly stopped to work. So yeah, a few major roadblocks.
Now that you mention, and as Sebastien mentioned at the beginning, that all details are usually lost in the very brief materials and method sections of most papers, because they don't specify, or they might specify the catalog, but do not elaborate on the fact that if you change provider, the results could change. And those could be months, if not years of work, trying to figure out, what changed from one day to another one. And it could be just the provider, of the materials. And what then would you say to the user are the most critical steps to undertake your protocol successfully?
I can try. So I would say the first thing is that it's really important to do conjugation on a solid surface, as Antoine mentioned. And as soon as there's any liquid left in the wells, for example, on the agar plugs, this really reduces the conjugation efficiency. So the delivery of the transposon is really affected by the presence of liquid. So it's important to dry the plates very well and make sure that the cells, when they're deposited on the plug, that all liquid is really absorbed so that the cells can get into very close contact and have very high conjugation efficiency.
Yeah, and it's not important, it's critical to the protocol. I don't want to understate how critical this step is, because like 10 minutes of drying the plate can be the difference between, can be like 3 log more insertion after 10 more minutes of drying the plate. So it's the difference between having to redo the experiment and having it successful. So really the critical step is the drying of the plate and the conjugation.
And since this protocol will be or is the foundation of many research projects, how versatile it is, how applicable it is to different bacterial strains. And that leads me to the second question, which is, have you had to troubleshoot or to optimize the protocol further since the time of publication, considering that you want to apply it to many different research projects?
So most of the projects that we're currently working on are based on E. coli. So if anyone uses E. coli, I think this should work pretty well. Now there are obviously many other bacteria that people work on, and I think the key is really to optimize the conjugation, the conjugative transfer. So if conjugative transfer has a very high rate, things will work well for the rest of the steps. But the key is really optimizing conjugation, and that can be done, for example, by changing the ratio of the donor strain compared to the target, the time of conjugation, the duration of conjugation, the temperature can also be optimized. So there are a few parameters that can be played with so that the efficiency is high enough. And if efficiency is high enough, the rest of the steps will usually go pretty well.
And since the publication of the protocol, we started a few collaboration with other labs, and we've tested a clinical E. coli strain where the protocol worked flawlessly as is. We've also tested conjugation toward Chronobacter sakazaki, if I recall correctly, another bacteria, and it also worked really good. And I don't know if it has been confirmed yet, but we've changed the cargo of the transposon, which consists of a resistance gene, to work with a collection of Bacillus subtilis strains. And we've sent this new construct to a collaborator, and we expect to hear from them really soon. But I think the
one key point that we didn't mention, but the conjugative plasmid that we're using is known to have a very broad host range. So it's not something that's really specialized for E. coli. It's an INC-P conjugative plasmid, or RP4. Some people will know it under this name of RP4. So it's known to conjugate well to a very wide spectrum of bacterium. Even yeast and even human cells have been used for conjugation with this system, though at lower rates for human cells and yeast. But that really shows how versatile this tool can be.
No, please, Antoine.
Since we designed the whole system from basically scratch, we've designed it to be easily adaptable toward other organisms. For example, if you need to change the promoter for an organism where the one we use don't work, or the resistance gene, we've made it so that it's easy to adapt to a new organism. Some of the system we used to employ for this kind of transposon mutagenesis were old and poorly annotated, and it was really hard to update them to current standards of microbiology. And with this system, it's designed to be upgraded and changed for the future. So
that's great. And regarding the promiscuity of the transposon, let's say, to other cell types beyond bacteria, if we think about the genome itself, is there any bias in insertion, or the transposon is equally promiscuous when we look at the genome itself?
So there's a slight bias, and it's been characterized in the literature, but the bias is so small that it's essentially random. So it's a TN5 transposase that we're using, and so some of the people that listen will know that this is actually used in the Nextera sequencing chemistry on the Illumina platform. So sequencing libraries are made with this because we assume it's random enough that we won't see that bias.
When we look at the sequencing data, we can detect the bias if we look really into it, but otherwise there is no apparent bias in the distribution of the insertion on the final data.
I think that the conversation we have had regarding the protocol itself, I think is going to be very useful, not only to the experienced researchers in this field, but also to the more naive or early career researchers. I now would like, just to close to the end, to tell me what has been the significance of getting this protocol, getting this paper peer-reviewed and published for each of your careers, considering the different stages of them, one being a professor and another one more a junior postdoctoral researcher.
I can start on my side. So being a professor, this really gave credibility. So when we submit proposals and we want to get some grants or money to perform projects, and we can say, look, we have this protocol, it's published, it works well, it's been peer reviewed, that really gives the credibility to more easily get the funding for the larger ambitious projects that we discussed. So that was the main advantage on my side.
Yeah. And on my side, I think it was like my first real scientific publication, so a milestone there. But it's also really an interesting publication because I could include like a lot of optimization and work that I spent a lot of time on it. So it's like a good way to show my work, and also to show not non-standard publication practices, because like it's a protocol which is like a recently developing way to publish scientific data. It also included like 3D printed materials which I designed. So I think it was a good way to share with the community that way. And it's also the base for many other projects that I'm working on currently. So publishing this protocol was like a milestone which allowed me to pursue other projects.
So you're making friends and new collaborators.
Yeah. Actually, it worked really well.
I'm really happy. I'm really happy to hear that. And now, before we really finish, is there anything that you would like to share that hasn't been mentioned and touched in the conversation?
Well, I think we really want people to use the protocol, and if people can refine it, or if they have any comments or requests, we're really open to working on this again and improving the protocol so that it's available to the wider community.
Yeah. And I would add that it was a great experience and a really interesting experience to publish the protocol as is and focus the research and writing effort on having the protocol being publication ready rather than shoving it in the materials and methods of an article. So it was both a good experience and I think a good thing in general for science. I think we should see more often protocol published in a standalone way. So, yeah.
Thank you so much. I'm really happy that it worked for you.
Yeah, thank you. Thank you.
Thank you.